It's pretty wild when you think about it. The tallest tree you've ever seen, the pet curled up at your feet, even your own body. It's all made of the exact same stuff at its core. So, what is that stuff? Today, we're going to dive deep and explore the fundamental unit that connects literally every living thing on the planet. I mean, it's a huge question, right? What is all life actually made of? If you could zoom in way, way down past what your eyes can see, what would you find? What's the one thing that's at the very foundation of it all? Okay, to even begin to answer that, we have to wrap our heads around scale. Just look at this chart. Our eyes are pretty good, right? We can see a chicken egg, no problem. But once you get down to the size of a human hair, and then even smaller, you're basically in a completely invisible world. There's a whole universe of things happening that we just can't see. So that's where we're headed today, into that world we can't see. It's a universe that's all around us, and even inside of us, and it holds the key to, well, pretty much everything about life. And the answer to that big question, it's the cell. Think of it like a Lego brick. It's the fundamental unit of life. Some living things, like bacteria, are just one single brick, but something like you or me, we're made of trillions of these bricks, all working together in this incredibly complex way to keep the whole system running. But wait a minute. If these cells are so small we can't see them, how in the world do we know they even exist? Well, to find out, we needed something revolutionary. We needed a new kind of eye. Here's the problem we were up against. 0.1 millimeters. That's the limit of what the human eye can resolve. What that means is, if two tiny dots are closer together than that, your eye just sees one blurry dot. And since almost all cells are way, way smaller than this, they were a complete secret for thousands of years. And then this happened. Say hello to the hero of our story, the light microscope. This incredible invention uses lenses to blow things up, to magnify them, letting us smash right through that 0.1 millimeter wall. For the first time ever, we could actually see the building blocks of life. It all started way back in 1665. A guy named Robert Hooke was messing around with a microscope he built himself. He put a super thin slice of cork under it, and what he saw were these little box-like compartments. They reminded him of the small rooms monks lived in, which were called cells, so that's what he names them. And just like that, a whole new science was born. It's amazing that the same basic idea is still used in labs everywhere today. All right, so we can finally see a cell. That's awesome. But what are we looking at? The first and maybe most important feature is its outer edge, the gatekeeper. This is the boundary that separates everything alive inside the cell from the chaos of the outside world. This gatekeeper is called the cell membrane, and honestly, the best way to think of it is like a bouncer at a super exclusive club. It's selectively permeable. That's the sciencey term. It just means it's super picky. It lets the good stuff in, like water and nutrients, but tells all the bad stuff, like waste, to get lost. This control is everything. Without it, the cell couldn't survive. So how does this bouncer actually do its job? Well, you can see it for yourself with a super simple experiment. Seriously, you could do this in your kitchen. All you do is cut a potato into two identical pieces. You pop one piece in some plain old water and the other one in some really, really salty water. Then you just hang out for an hour and see what those cell membranes decide to do. And check it out. The difference is huge. The piece that was in plain water looks all swollen and puffy, but the one from the salt water, it's all shriveled up. So what is going on here? What you're seeing is a process called osmosis. It's all about balance. See, the potato cells want the amount of stuff, in this case salt, to be the same inside and outside. In the plain water, there's more salt inside the potato cells, so the membrane lets water rush in to try and even things out. That's why it swells. But in the super salty water, there's way more salt outside, so the membrane lets water rush out of the potato to try and balance that. And that's why it shrinks. It's a perfect visible example of that selective gatekeeper doing its job. Okay, so every cell has that membrane gatekeeper. Got it. But that doesn't mean all cells are the same. In fact, there's a huge difference we can see when we look at a tale of two cells, plant cells versus animal cells. And wow, the difference is just obvious, isn't it? Look at the onion cells on the left. They're so orderly, like little bricks all lined up in neat rows. And then look at the human cheek cells on the right. They're kind of all over the place, totally irregular, no fixed shape at all. So why the huge difference? It comes down to one simple but really important thing. Plant cells have an extra layer on the outside, something called a cell wall. It's tough, it's rigid, and it's what gives plants their structure. Animal cells, we don't have that. And that lack of a rigid wall is exactly what lets our cells be flexible, which we need to do things like, move our muscles. So the light microscope opened up this whole new world for us. But you know how it is. The more you see, the more questions you have. We wanted to see even deeper. And to do that, we were going to need a much bigger boat. Or, in this case, a much more powerful microscope. First, though, let's give the light microscope its moment. This thing was a complete game changer. It literally revolutionized biology. It was the key that unlocked that first door to the invisible world. But there were so many more doors to open. And for those, we needed a new key, a master key. And that is the electron microscope. So here's the crazy part. Instead of using light to see things, this beast shoots a beam of electrons. The result? A level of magnification and detail that is just mind-blowing. We're talking about seeing tiny little structures inside the cell that someone like Robert Hooke couldn't have even dreamed of. And this is what that power gets you. This is not a drawing. This is a real photograph of the surface of a leaf, taken with an electron microscope. The detail is just incredible. It really shows you how every time we build a better tool, we uncover a whole new layer of reality. All right, so let's quickly recap what we've covered. We started with the big one, what's life made of? And the answer is cells. We learned that technology, especially the microscope, was the only way we could ever discover this hidden world. We saw how the cell membrane acts as that all-important gatekeeper, and finally we saw that a simple cell wall is the key difference between plant and animal cells. But here's the thing. We've really only just scratched the surface. It's like we've seen the outer walls of a giant, bustling city, but we haven't even stepped inside the gates. Which leads to the next big question. What kind of incredible machinery is humming away inside these tiny building blocks of life?